Anyway, so I'm going to pick up a little bit on what some of the things that Ron introduced, and also draw upon some of the work that you've been doing in the lab with the microfluidics, and also take the opportunity to tell you just a little bit about what Bill and Sebastian and Jordi are doing in their work. So we're going to be talking about microfluidic technologies, and you've heard a little bit about EBICs off and on. I want to tell you a little bit more about that and how emergent behaviors in biological systems relates to developmental biology, because it really is, development really is all about emergent behaviors in biological systems. So this is kind of an overall roadmap of what I'm going to go through. It's a little disjointed. I want to cover a number of different things, but I think there is kind of a thread to it. So just starting out in terms of basic background, what are we interested in EBICs? EBICs, by the way, remember, is emergent behaviors of integrated cellular systems. So development is something that would fall under the purview of EBICs. So what we're looking at is cell populations, and what we find is that these cell populations exhibit various types of what are called emergent behaviors. So you might start out with something like this, which is this happens to be a collection of cells which are expressing two different types of surface adhesion receptors. So the green expresses one type of receptor, the red expresses another type of receptor. You let these go for a while, and they self-segregate. Okay, so that they sort of reach a minimum energy state, so that the cells that are expressing the strong adhesion molecules segregate to the inside, and it leaves on the outside a ring of the cells that are expressing the low adhesion molecules. So here's a case where you get pattern formation from a very simple process. But it's through the interaction between the individual cells that this separation is occurring. Something a little bit more complicated is if you go to systems such as this, where you're starting with an endothelial monolayer. So what you're seeing here are the blue is a DAPI stain for the nuclei, the green is an actin stain. So what you're seeing is the formation of a vascular network down into a three-dimensional gel. So here again, it's a complex network that's being formed seemingly by itself, but it's through signals with neighboring cells, probably as a result of sort of global signals that the cells are receiving, that gives rise to some sort of an interesting pattern. So the research that we're doing in EBICs, there's our logo down there, is really aimed at trying to understand how these emergent properties arise, okay, and how ultimately we can control them. Because what we'd like to do is be able to basically replicate what you do in development, developing a new embryo, developing a new organism, but to do it by our design. So we want an organism that has a certain function in the end, that doesn't exist today, that's not in nature. We don't want to simply reproduce what nature is already doing very effectively. We want to use nature's tools to produce essentially new organisms with new functionalities. A lot of the work in our lab has to do with formation of vascular networks, because if you develop an organism that's more than, say, maybe a millimeter in dimension, that organism is going to need some sort of a circulatory system. So, we're sort of looking down the road a little bit. The systems that we're developing initially are minute in the sense that they're small enough that they wouldn't need the circulatory system. But we're anticipating going up in scale to the point where a circulatory system would be necessary. So we've had a lot of discussions. In fact, Ron and I and a few other people in EBICs have been meeting on a regular basis really to try to define what we mean by emergent behavior in the context of what we're doing with our center. And we've come up with this definition. And it may seem straightforward, but it's actually a result of a lot of discussions that we've had. So first of all, it's self-directed. In other words, the cell population itself is really determining the ultimate behavior that you see. It's a multicellular response occurring as a result of collective interactions of individual cells. So for example, each individual cell behaves in a certain way. It expresses certain receptors, has certain adhesion molecules, has certain signaling pathways that are activated. And if you were to look at that single cell, you wouldn't be able to, at least at this stage, predict how it would behave when it's interacting with a population of similar cells. Okay? So one of the keys is that we really have to look at a cell population in order to determine what this emergent behavior ultimately is going to be. So what determines this is in part the local microenvironment that the cell senses, and that manifests itself in phenomena at macroscopic scales. So I've pictured here a couple of things that you think of as emergent behaviors. Formation of tiger stripes, the vascular network that I just talked about, patterns on a butterfly, and here this is a school of fish. So it really happens at all different levels, all different scales. It turns out that there's a really interesting history in discussions of emergent behavior that go back probably 100 years. People have been talking about it for a long time, and people have different perceptions about it. But we're trying to focus in on what it means in the context of biological systems as opposed to other non-biological systems, like things like Turing patterns and things that you probably have heard about. So we like to think of there being really two ways to develop what you can think of as a biological machine or an organism. One is the way that nature does it. So this is a collection of stem cells. You can imagine, an inner cell mass ultimately evolving into, in this particular case, into a zebrafish. Okay, and you can imagine all the different types of behaviors that have to happen just right in order for the zebrafish to develop. And it's really kind of remarkable that given all the stochasticity, we talked about earlier in terms of, the fact that biological phenomena are so stochastic and there's a lot of randomness and variability. But despite that, a zebrafish always results, okay? So what is it that kind of takes all this noise and reduces it so that you have a very consistent end product? So this is one pathway to develop an organism, but you can imagine too, we could take these stem cells, we could differentiate them into neurons, into muscle cells, into endothelial cells individually, and then we can put them together. So we can make organs, we can make liver, we can make kidney, we could make, say, an organ that senses the level of a particular drug in the blood, instructs another set of cells to synthesize that drug, and then secretes that drug into the body in order to maintain the level of the drug constant over time. Say, for example, if somebody has a chronic illness and requires constant medication. In fact, one of the machines we're looking at is what we call a beta cell factory. Basically, it's developing a pancreas that could be inserted subdermally that would sense glucose levels and then instruct beta cells to produce insulin so as to maintain insulin levels constant in a patient. So that's the kind of thing that we're looking to do. To do that, it really, we've argued that it requires development of a new discipline because it's really a combination and draws upon all these different things that you see here. You just heard Ron's perspective from synthetic biology, but there's also systems biology. Systems biology has sort of different connotations. It's come to be known as sort of what's happening inside the cell and kind of the genetic processes that occur. But here I mean systems biology in a much broader sense, sort of multi-scale sense, where we're looking at really complex biological systems that go from single molecules up to cells, up to cell populations, and ultimately up to organisms. Mechanobiology plays a role. We've seen a lot from Ray Keller's talk, for example, and also Lance's talk and Larry's talk yesterday, where the mechanical processes, the mechanical interactions between cells that occur in an embryo really, to a large extent, determine how that embryo develops, how it evolves. We're finding that control theory is very important. There's a lot of feedback that goes on. Okay, each cell is sending out signals, but the behavior of that single cell depends on the cells that it's interacting with. Because it sends signals out to other cells, it modifies the way in which those cells are behaving, and then those cells in turn send signals back to the original cell and modify its behavior. Okay, so you can think of that in the context of sort of traditional control theory that people have used, and Harry Asada in our department is very interested in applying control theory to biological systems. Tissue engineering is kind of an interesting history. You know, I think it was Time magazine that back in 1990, I think predicted that tissue engineering was going to be the profession of the 21st century, and made all sorts of predictions about the size of the business and everything else, and really none of it came to fruition. It's starting to now. I mean, there are actually products on the market, you know, that are tissue engineered products, but it really hasn't developed as extensively as people predicted 10, 15, 20 years ago. And part of the reason is that, you know, we tried to do it, you know, we thought we just put all these cells together, they'd automatically know what to do, and they'd form these organs, and then these organs could be implanted. But in fact, what we're finding is that you really have to understand what's going on. It's not sufficient just to put these things together and they're going to do their thing, but you really need all these other parts. And then also, you know, you've seen a little bit of numerical simulation along the way. And numerical simulation, I felt that Larry was a little bit pessimistic about, even though he does numerical analysis primarily. I see a numerical model as sort of a receptacle for what you learn from experiments, and that it's a constant iterative process. So, you know, you learn something from experiments, you use that to inform a model, you make predictions from the model, you form hypotheses using the model, you test those hypotheses experimentally, and then you find that, well, the model doesn't quite fit, so you have to go back and then sort of adjust it. Okay, and there's a constant iteration, constant back and forth, but as that model develops and becomes more complex, it can actually capture much more behavior than you can think of, than you can keep in your mind at one time. So especially with biological systems that have such high complexity, you know, I think the computational aspect of it is extremely important. So if you put all these together, you know, we're trying to use that then to develop these integrated cellular systems, or the biological machines. So what do we need to do in order to get there? This is sort of a slide of kind of a roadmap of first of all, starting with stem cells, how to control stem cell differentiation, say either in isolation or in an embryonic body or in an embryo. So it controls differentiation both in isolation and as part of a population of cells. We have to understand how cells communicate. Okay, we know a lot about cell-cell signaling. Okay, we know a lot about the signaling pathways inside cells, but what we need to understand better is how this communication leads to coordinated behavior. In other words, when we have a population of cells, how do we think of this population of cells as being like a module or a component of one of the machines? So it might be a muscle, it might be a secretory organ, it might be a sensory organ, it might be something that processes information. Okay, and these modules then, we have to go up one level of complexity and then assemble these modules to produce the functionality of some sort of a biological machine. What I can show you, and maybe just a little bit in the talk today, but we pretty much have control over these. I think we still have a long ways to go before we can really assemble these in an effective way and have something that's functional, and not just functional for, you know, for an instant in time, but functional over a long term. Because the stability of these organisms or organs or whatever they are, the long-term stability is probably one of the major challenges that we face. Because the cells tend to start to de-differentiate once they form the structure. And we've talked some about the technologies. We've heard a lot already about imaging technologies, you know, deep imaging of signaling events, real-time, high resolution. You're going to hear more about that. Peter So is going to speak, and Daipan, are you going to speak as well? Yeah. So imaging obviously is critical in terms of trying to observe what's actually going on and then trying to use that to understand the processes. And then ultimately, we had to have new ways of controlling and measuring the forces and the signaling on a microscale. Again, the forces, the mechanobiology aspect is critical. And then finally, new ways of regulating sort of the external or global macroscale phenomena or signals that are being received. The environment that the cells are in, growth factors, oxygen partial pressure, pH, temperature, electric fields. Being able to control these in sort of a macroscopic way for either for an embryonic body or an embryo or a collection of cells is going to be critical in being able to develop these machines. And this is really where the microfluidics comes in. So we've talked about, this is actually a slide I borrowed from Todd McDevitt's presentation, where you can sort of think of the development of the gastrulation being replicated by phenomena that are occurring in an embryonic body. Remember, an embryonic body is simply really an aggregate of embryonic stem cells that then start to differentiate into the different levels, the ectoderm, the endoderm, the mesoderm. And in an embryonic body, when we form these, and I think you're seeing this probably in the lab this week with the, we're going to look at the differentiation into cardiomyocytes in an embryonic body. The differentiation tends to be kind of random, okay? But these things naturally want to differentiate into different cell types, okay? So you see some cells differentiating into myocytes, some into bone, some into different cell types, neurons, for example. Okay, at this point, all we can do is say, yeah, this differentiation occurs, and we get these different cell types and the different cell layers, but we don't know how to organize them. We don't know how to apply external signals in order to get it to do something like what happens during development. Okay, and part of it is that we don't know how to control these things. We don't know how to control, say, the macro environment in such a way that we can get this to do the kinds of things that this does. And I mentioned that one of the things that we're very interested in is how do vascular networks develop. And I just wanted to define some terminology initially that typically in the embryo, what you see is what's called vascular genesis, where you have vascular progenitor cells that differentiate and at the same time develop into a primitive vascular network. As these develop, and either during development but also during repair processes later on, or in the case of, for example, somebody has a heart attack and they need to develop a new vascular system to help perfuse the region of the heart that's been damaged, what you find is that the same process can be reinitiated, where the endothelial cells then will recruit other cells like parasites and pericytes and smooth muscle cells to develop new vascular networks. And most of our work has been done really looking at angiogenesis, and this is in conjunction here with lymphatic vessels as well, and the same thing can take place in lymphatics, of course. But we're also interested in some of our experiments looking at vasculogenesis. So in some of our simple machines, if you think of a small scale, we're looking at things, this is just one example, where this happens to be the open circulatory system of an insect, where what you see here are neurons that are activating skeletal muscle cells that have organized themselves in a helical fashion around a blood vessel. Okay, so it's like an active blood vessel that pumps fluid or blood around the insect, around the circulation. So probably some of the early machines that we developed, this is the kind of circulatory system that we would use. And we can start to think about developing these in a system like this where, how many of you have heard of methods or seen papers where you can use an inkjet printer to print down cells in a certain pattern? Okay, so you can start to think about, here for example, we could have an inner layer. We could build this up through three-dimensional printing with an inner layer of endothelial cells, an outer layer that might have neurons that would activate, and then a muscle layer which would allow us to produce some sort of a pumping action in this vessel. Obviously there need to be some sort of a check valve too, a one-way valve in order to maintain flow in one direction, or it could be a peristaltic wave, okay, that squeezes along and pumps the flow in one direction. And this is meant to indicate, you can activate these either by motor neurons, but the question is, well, how are you going to control those? Well, one way is to do it optogenetically. Optogenetics is essentially what you do is you reprogram cells so that they express a transmembrane channel that is activated by light. Okay, so Harry Asada here has done it with muscle cells. Ed Boyden at MIT has done it with neurons. You could imagine a system here where we'd send a wave of light along the vessel, which would lead to a wave of contraction, which ultimately could lead to pumping. Now, obviously it's not very practical in the long term. We'd like to be able to generate those signals separately, and that would be, say, some sort of a processing unit that we'd also develop. But this is the kind of thing, long term, that we'd like to develop. So what I want to do is, I'd like this to be interactive. So I thought it would be useful. You've done some microfluidics. You know basically what microfluidics can do. How can microfluidics be useful in developmental biology? Okay, what kinds of things could you do with microfluidics? Of the things of the type that you've already seen that might be useful in developmental biology, either in terms of understanding it, or controlling it, or manipulating it. Any thoughts? Control the environment. Okay. So what? And when you say that, whoops. What aspects of the environment would you want to control?
Gradient.
Okay, gradients. And what kind of gradients? Okay, chemical is one. Others? Temperature?
Pressure.
Pressure, okay. So that would have, and I guess I could say, that would control two things. One, it could control sort of the forces that the cell is experiencing. But it also, if we have, for example, the cells immersed in a matrix, the pressure gradients would give rise to interstitial flows. Okay, and I'm going to denote interstitial flows just by IF, so I don't have to write it out all the time. Okay, so gradients would give, and let's see, anything that we've missed here? I mean, you could also imagine gradients in oxygen tension, gradients in pH, electric field gradients. There's a lot of work being done in developmental biology, in fact, in terms of understanding how electric fields and electrical stimulation, local depolarization of cells, can lead to different patterns of behavior during development. A guy by the name of Michael Levin at Tufts is doing a lot of work on that. So, control the environment. We've got gradients. Anything else you might want to control?
Separate cells, maybe.
And what?
Separation of.
Yeah, okay. Let's say separation and organization of cells. So, for example, it's probably necessary in development or in making a biological machine to have the cells in the right location relative to one another. Distances are important so that they're close enough so that they can signal. If they're interacting through force, they have to have some sort of an arrangement. Are they on a monolayer? Are they in 3 dimensions? What kind of matrix material do you have? Whoops. In fact, in terms of matrix, usually we grow the cells either on 2-dimensional surfaces where we'd have some sort of adhesion receptors, or we might seed the cells in a 3-dimensional hydrogel, say, in which case, the biochemical nature of that matrix and its mechanical characteristics both would play a role. So I'll say both mechanics and chemistry And what I mean by chemistry here is that we can functionalize the matrix with different molecules that will control or influence the behavior of the cells that are suspended in it. Okay, you've probably heard of various types of hydrogels that people use. PEG gels are very common in terms of being very designable, manipulable. You can functionalize PEG gels, but at the same time, things like Matrigel, which is derived from biological materials, is used quite a lot in cell culture. Collagen is used very frequently. Fibrin gels, all these hyaluronic acid gels. So the matrix and both the stiffness of the gel, and you saw a little bit of that from Jimmy's talk, and the chemistry are both important. Anything else?
Like the orientation.
The
Orientation.
orientation. And by that, do you mean like the polarization of the cells, or? Okay, so like for example, during gas relation, we saw how important it was that you had the cells organized in such a way that there was an apical side and a basal side, and the behavior inside the cell was different depending on how those cells polarized and the wedge formation. Anything else? I think these cover most of the ones that I was thinking of. So good. So next, so let's move on to some of the microfluidic technologies and see, if we'd like to control all these, how many of these can we actually do realistically in a microfluidic device? This I find kind of interesting. It was taken from an article, it's just down here, 2009. There we go. Where it was mapping just the number of papers in PubMed that have the word microfluidics. And notice that prior to 2000, really microfluidics wasn't even around. At the time of this paper, in 2009, it had gotten up to about 100 citations a year. I just looked at it 2 nights ago. So in 2012, and that's just for part of the year, just for 6 months, the number's already at about 300. Okay, where the total number of citations, sort of integrating this curve, is up to around 5,000. So, it looks like things are leveling off, but in fact, we're way up here somewhere still, and still growing. So, microfluidics as a technology, I think, is still, I think, it's not nearly mature yet. There's a lot that really needs to be done. So there's a lot of opportunity. Some of the early microfluidic systems, this is one that a lot of people have heard about and a lot of people have used in terms of, we've talked about developing gradients and looking at chemical gradients as a means of guiding cell migration. George Whiteside's lab, and this was a paper, Nuli Jung was in George Whiteside's lab at the time that this work was done. And all that they did is they developed this serpentine pattern upstream. They put some, say in this case, a blue dye in input A and a clear dye in the bottom. And by the time it gets down here, after going through this pattern, what you have is a nice, well-defined gradient, chemical gradient. Okay? So that's been used quite a lot for people who are looking at chemotaxis. Migration on a two-dimensional surface under the action of a biochemical gradient. Here's another way to do the same sort of thing. In this case, they put one concentration of a chemoattractant here, different concentration down here, and these are actually separated by regions of agarose gel. And then the cells are plated in a central channel, and then you can watch the migration. Another system, here's one where there's a sandwich region of hydrogel. There's an upper layer where you have 2 reservoirs. So you have a low concentration reservoir here, high concentration reservoir. You have a gel that goes between the 2 reservoirs. And then underneath that are fluidic channels where you can actually, that are subjected then to the gradient that's established in the gel layer. Here's another system. Here's one again, chemical A, chemical B, and then in these channels between, which are small enough so you don't get a convective flow through it, you can generate a gradient. And I know in the lab you generated a gradient yesterday, and you know how difficult it can be to get a nice gradient. Everybody experienced that, I think. And these are all ways in which people have tried to do this, just and not even within a gel. All these that I've shown you so far are simply in channels. Okay, so the cells are migrating on the two-dimensional surface. Well, we also talked about scratch assays. You know, back at Jeff Redberg's talk, remember he was looking at the migration of cells from a monolayer, and they did the scratch assay. They sort of wiped out the cells on one edge, and somebody was asking the question, well, you know, what are you damaging the cells on the edge? Is that influencing the behavior of the cells? Well, here's an experiment that Mike Morrell did a couple years ago and just published last year, where what he did was he produced the same type of wound, essentially, by introducing trypsin that just lifted the cells off the substrate rather than killing them. Okay, so he could actually do this in a way that was non-damaging to the cells, and then he looked at the migration of the cells. Here what he did is he actually introduced hydrogen peroxide in a gradient, okay, by introducing it into one of these channels and then regular cell media in the other two channels. So he produced a gradient in cell death to sort of simulate the effect that you see if you physically scratch the surface. So he was looking, and you know, I won't go into the details of what his findings were, but that, you know, there were differences due to the fact that in the traditional scratch assay, you actually do damage the cells, you kill the cells on the edge, and if you simply lift them off, you get a somewhat different response in terms of their migration behavior. And this is just a collection of some of the different types of microfluidic systems that people have had. I thought it'd be useful for you to have this slide. This has a lot of the references, so I can't say which reference connects with each individual picture, but here are some that have occurred, and most of these, I guess the oldest one dates back to about 2002, so you can sort of see how the field has evolved over the period from 2002 to in this case, to about 2008. A lot of them, so those are all in sort of two-dimensional migration assays. And you know, there's a lot of microfluidic work that's also done in terms of single molecule analysis. You know, you can do DNA in a chip, PCR in a chip, so single molecule. You can do single cell sorting on chips. There's a lot of interest in that, especially in the context of things like separating circulating tumor cells from the blood, these rare cells that just about any cancer patient has in their blood. There might be 100 of these in total in a milliliter of blood, so they're extremely rare. But if you can separate these out, you can imagine you can do all sorts of diagnostics on those cells and see what you need to do, what agents you need to provide, drugs you need to provide to the patient in order to prevent these cells from actually recolonizing and forming metastatic tumors. So, so that's all. A lot of that work is in microfluidics. In this case, we're using, this is not a microfluidic system, but it's a kind of a traditional assay for monitoring cell migration across membranes or even through three-dimensional matrices. And this is the product that you buy, and what it is basically is a well that has a porous membrane on the bottom that separates two regions of medium. And typically what you do is you plate the cells either directly on top of this membrane, or you can put a layer of gel on top of the membrane and then put the cells either in the gel or on top of the gel. Some people have done co-cultures where they plate a different type of cell either on the bottom of the plate down here or on the bottom of the membrane, so you can look at interactions between the two different cell types. And what they typically do is they'll let this assay run for a while, and then they'll take it out. They'll detach the cells, in this case using trypsin, and then count the number of cells that have made it through, either from the top of the membrane to the bottom or through the gel to the bottom of the membrane, as a migration assay. Okay, so it's nice in the sense that it's a relatively simple system to use. It gives you a measure for the number of cells that have migrated through, say, a gel of a certain thickness over a certain period of time. So it gives you a quantitative measure of migration. but it doesn't really allow you to watch things. You don't really see very much. You can't image what's going on during the course of the experiment in a transwell system. And that's where microfluidics, I think, has a big advantage. So the system that you've been using is kind of a variation on what's shown here. In this particular case, instead of just two channels, this one has a central channel as well. The pink regions here are regions where we have introduced the three-dimensional gel, the hydrogel, either with or without cells mixed in. The three channels here are accessed from the top of the device. This is a device like you're using, which is about that so big. And this just shows how different types of seeding procedures that you can use. Seeding cells in different cells in different channels, either with or without cells in the gel region. You can do the simultaneous 2D and 3D culture, 2D in the channels or 3D in the matrices. You can image things well because everything happens within 100 or 200 microns of the cover slip. You can do multiculture. In this case, you could imagine seeding up to 5 different cell types. That's too complicated for us right now, so we're mostly working with 2 and maybe 3 different cell types at most. But then some of the things we're talking about here. In fact, we didn't put in different cell types. Control the environment. Let me add that here. So co-culture. Because obviously you have different cell types that are interacting with each other to produce the different types of behavior that you see. Concentration gradients, flow, and hypoxia. All these things, in principle at least, you can generate in a microfluidic system. And these are some of the different arrangements that you can imagine. This is from one of our recent papers. And here you can see cell seeding. For example, in this case, if you want to look at migration through a three-dimensional gel, you can seed the cells in the channel. You could set up a concentration gradient, say a growth factor gradient, and then you can observe in real time the invasion of these cells into the gel and the migration through the gel. Here's a case where you can introduce epithelial cells in one channel that form a monolayer, either epithelial or endothelial cells, which would then subsequently sprout depending on what factors are introduced in the other channel, which would be over here. We've done this looking at tissue-like three-dimensional structures where we might seed the cells in a fairly dense matrix on the side surface of the gel. That some of the cells might break loose, and you might look at, for example, things like metastasis of cancer cells or EMT. We also use it to develop sort of hepatic-like, liver-like tissue layer on the surface, and then maybe interacting with other cells on the other side. Here's a case where we embed cells inside the gel. One set of experiments that we're doing is we form a spheroid from cancer cells, cancer cell line, a spheroid which you can think of it as a small tumor, which we can then suspend in the gel, and then either with or without an endothelial monolayer there, we can introduce drugs to the channel to see what drugs can actually limit the process of EMT, the separation of the drugs away from the primary tumor and to migrate through the tissues. And you can do this then, as I say, looking at interactions with an endothelial cell layer, and in fact you can look at the intravasation process. The cell is not only migrating through the tissue, but also coming up to the endothelium and then transmigrating across the endothelium to get into what would be the vascular system in the patient. You can look at floating cells. We've done some experiments with bacteria, for example. You can imagine setting up chemoattractant gradients and then looking at the migration of bacteria. Or if you have an endothelial monolayer here, you might be looking at adhesion to that monolayer of neutrophils or circulating tumor cells, and then the extravasation of the cells into the matrix, and then co-culture. Different types of different cells that we might introduce. So all these are different ways in which you could use essentially the same kind of device. So some of the things that we've done, I mentioned, I'll come back to this more, looking at angiogenesis. These are vessels that are grown from an endothelial monolayer that are sprouting into a three-dimensional gel. Here we're looking at interaction between endothelial cells that are plated in the central region. These are human microvascular endothelial cells. The white rectangles correspond to where the gel is located. And then over here we have what are called 10T1/2 cells. It's a precursor to smooth muscle cells. And they can differentiate into pericytes. And the idea here was that we wanted to look at the interaction between vascular sprouts and these pericytes, which would help to stabilize the vascular network over time. Another experiment, this is done with a collaborator in Japan, Ryo Sudo, and also with Linda Griffith's lab, where we introduce liver hepatocytes. We sort of pack them in on one side of the gel. We introduce endothelial cells on the other side, and here you can see the endothelial cells that are sprouting through the gel. They're heading toward the hepatocyte mass. What we found was that these structures, these vascular structures, would come right up to the hepatocytes, and then they would either stop or they would turn around. What we were hoping to do is actually get these vessels to penetrate into the hepatocyte mass and start to develop something that had function more like liver. But then we looked and thought, well, during development, what happens is you have these hepatic stellate cells that actually are sort of the sentinels that lead the endothelial cells into the matrix, into the hepatocytes. So what we did is we introduced the stellate cells into this channel at a later time point. They did migrate through the hepatocytes, and they're intermingling with the endothelial cells here, but in fact they still didn't cause the endothelial cells to get into the tissue. Some of the later work that Ryo's done, actually, he's getting better success now at getting the vascular networks to grow into the tissue, sort of going toward sort of an engineered liver. And some of his students and postdocs are interacting with our group to do that. Here we are looking at axon guidance. There's a lot of interest in terms of, in spinal cord repair, to get the neurons, the end of the neurons on one side to grow across a region of damaged tissue and connect with the neurons down here. So you're interested in identifying what the chemoattractants are that actually guide the axons. Well, in this case, what we've done is we've used a known chemoattractant, SLIT2. This actually tends to be a chemorepellent. So in fact, we've introduced the tumors or the neurons down here. They send axons up into the gel region, and then they tend to be tilted away. They're repelled by the slit, and they tend to migrate a little bit more toward the left. So we can test then as a function of concentration. We can look at different chemoattractants or chemorepellents to see what might be useful in terms of promoting axon guidance. Tumor cell migration, I'm not going to say much about this, but a lot of the work in our lab has to do with cancer and studies in terms of looking at metastatic cancers. In this particular case, this is a cancer cell line which is invading through the gel. This is a three-dimensional collagen gel, coming up to an endothelial monolayer. What you can see here is a VE cadherin stain along with a DAPI stain. And these cells are actually transmigrating across the endothelium, getting into the vascular system, and actually could then promote remote tumors, could cause metastasis. What we found in these studies is that it's really tough for tumor cells to get across an intact endothelial monolayer. That of the cells that get up to the endothelium, only a relatively small percentage of the cells can actually get across. What helps is if you then co-culture these cells with macrophage. Macrophages tend to, again, sort of promote the migration of the tumor cells to the endothelium and then transmigration. So the percentage of cells that can transmigrate actually tends to increase by at least a factor of 2 and probably more. And here's a case where we've just grown epithelial cells on the surface. Those epithelial cells form these nice ductal structures. Again, you can replicate some things that happen in vivo. And I mentioned that you can start to do this in high throughput. Your devices just have a single region, experimental region that looks something like this. We're now developing systems where we can multiplex it and do more experiments in a shorter period of time. So in view of that, okay, what capabilities would be useful in the use of microfluidics for developmental biology? Well, it's pretty much the same kinds of things that we were talking about here. What capabilities would you like? Well, you'd like to be able to generate gradients, all of these types of gradients, generate flows, control the forces on the cells, you know, separate the cells, organize them in certain ways, use different matrices, okay? All these things still apply, okay? So focusing in on microvascular networks. What I'm first going to tell you a little bit about is some work that that a student, Vernella Vickerman, in our lab did, where we're looking at the interaction between interstitial flow and the formation of vascular networks. So the device is very simple. It's even a little bit simpler than the device that you guys have used in lab, but it's somewhat harder to use. And the problem here is that in order to be able to control the flow through the matrix, we didn't use the gel filling channels. So instead, the gel was micro-injected into the system, and then the cover slip was subsequently added on top. It has some problems in terms of getting the cover slip to bind properly, but those problems can be sorted out. We can get the system then seated with cells, endothelial cells on one side, generate a pressure difference across the two channels, and then generate an interstitial flow. Now, in your case, when you're forming the gradients, any interstitial flow that you formed that generated as a result of, you know, tipping the device or moving it around messed up your gradients. Okay, in this case, we're actually purposely generating a pressure differential just by means of changing the pressure at these ports so that we can control the interstitial flow through the matrix. And had sort of an interesting effect. First of all, this is the kind of thing that you can see. This is an endothelial monolayer on the top. This is the channel out here, and this is the hydrogel. And what you're seeing is the formation of these vascular structures diving down into the gel. It's in 3D, and we're only focusing on one plane. So, you know, some of these things are out of focus. Others are a little bit more in focus. And in this particular case, using the combination of vascular endothelial growth factor and sphingosine 1-phosphate, you can see here, you can see the formation, beginning formation of a vascular lumen. And we're now able to get these actually to grow all the way across the gel region and start to perfuse them. In Vernella's studies, what she did is she wanted to see what the effect was of, if you have a monolayer here, what happens if you drive a flow in this direction as opposed to if you drive the flow in this direction. So the channel is out here, the gel is down here. And it's kind of the same situation that you have in the region of a tumor. So tumors, as you might know, tend to be a region of high interstitial fluid pressure. So what happens is you have pressure gradients from the tumor emanating outward, which would drive interstitial flow from the tumor toward either the lymphatics or the low pressure venous side of the circulation. So we were trying to simulate that here. You can imagine in this case, this would represent sort of the low pressure side of the circulation. So this might be a vein or a venule. This would be the tissue. The tumor might be down here. The flow would be generated from the tumor heading toward either the venous circulation or lymphatic. Here the flow is in the opposite direction, so this would maybe better simulate the arterial circulation, flow from the channel into the gel. And what Vernella found was that in all cases here, and this is with or without any additional growth factors to actually form vascular networks, that the flow actually gave rise to very active formation of vascular networks if the flow is from the tissue out into the vessel, and virtually nothing when the flow is in the opposite direction. And this goes back, there was an observation by Judah Folkman, who is the person who really developed anti-angiogenic therapies for cancer. He had made the observation a long time ago that the new vessels that form that eventually perfuse a tumor tend to emanate from the venous side of the circulation as opposed to the arterial side of the circulation. And at that point, it was really just an observation, but this could be the explanation for it in terms of the direction of the flow across the endothelial wall. So we wanted to see whether this was a function of we were using microvascular cells for those experiments. we thought, well, what happens if we use these HUVEC cells, human umbilical vein endothelial cells? Well, exactly the same thing happened. Got the same response. So it seemed to be independent of the type of endothelial cell that we were using, but was sort of a general function of, or general behavior of endothelial cells. So we thought, well, in the system we can start to probe what's the mechanism by which this is actually happening. And we first of all thought, well, due to the flow, we're probably setting up some sort of a polarization of any growth factors that might be in the medium. Because if you've got a barrier here, you're going to get a pileup of protein on the bottom side of that monolayer, and that could be a gradient that could actually give rise to polarization and might induce growth. So, but then we just flooded the system with VEGF, with this vascular endothelial growth factor, and got exactly the same response. So although there are other growth factors that could have been involved, we thought, well, that seems maybe not the likely explanation. So then we turned to mechanical force. And if you think about it, if this is the gel down here and these are the cells that are attached to it, these cells, you saw focal adhesions, okay, from Mike's presentation and I think Chris's as well. Well, the focal adhesions, they're adhesion sites that tether the cells to the matrix. If the flow is going in the direction of the arrows, that means it's kind of lifting up the cells. It's trying to tear the cells away from the matrix. So the cells are holding on through these adhesion sites, which means that there has to be a fair amount of stress or force localized to these points of adhesion to the matrix. There's also could be stresses in the cell-cell adhesion junctions. In this case, it would be the VE cadherins at the cell borders. But from the experiments that Vernella did, it looks as though it's probably this mechanism as opposed to that. And the way we could tell that is by going in with different types of inhibitors. And specifically, anything that we did to, say, for example, block Rho kinase. Genistein here is a general tyrosine kinase inhibitor. PP2 is a Sarc family tyrosine kinase inhibitor. Here's the control case. Any of these interventions tended to reduce the formation of these vascular networks drastically from the control case. Interestingly, heparinase 3 also had a similar effect. Anybody have any idea why heparinase would have any effect? Heparinase is an enzyme that breaks down heparin sulfate. And heparin sulfate tends to be in the glycocalyx on these endothelial cells, which could be on the basal side, but is certainly on the apical side. Okay. Any ideas? Yeah, we didn't have any either. Except that there's another guy, John Tarbell, who's been doing a lot of work with glycocalyx. And what he's postulated is that there's actually a mechanotransduction connection between the glycocalyx and the endothelial cells. And that by removing the glycocalyx, you're removing the ability of the cells to sense force that way. And the glycocalyx, could be on the apical side, but it could also be on the basal side. So it is known that that it tends to inhibit angiogenesis, but how it relates to some of these other factors is still somewhat of a mystery. Okay, so in terms of force generation on the vascular networks, there's this work that I was just talking about. There's also work that Jordy's doing for his thesis, looking at the effect. Again, the monolayers on the upper surface here in both cases. This is a situation of a vascular network forming under static conditions. Okay, in other words, the pressure is equalized across the gel region. Here's a case where he has a transmural flow. In other words, the flow direction is the same as what you see here. It's going from the matrix out into the channel. But you can see the effect that it has on the formation or the morphology of the vascular network. We expected it to be the transmural flow to be something that would occur in vivo and that would probably lead to a better developed, more stable vascular network. From the initial results, it looks like it has just the opposite effect. We've tried the flow both from this direction down, in other words like this, or from this direction up. But it seems so far as though the static configuration, one where we don't have any flows generated, seems to be optimal. But those are still experiments in progress. There's been a fair amount of work in terms of, for tissue engineering especially, how do you engineer microvascular networks? Here's some work that's been done by Chung Kim in our lab. And what he's trying to do, again, is reproduce this situation where we have growth factors that are being secreted by either cells, or in this case, from cells that are encapsulated in beads that could be implanted into the body or used in a microfluidic device like this, that are releasing things like VEGF that would then induce a vascular network to grow from existing existing vessels. In order to do this in co-culture and to do this stably, one of the problems you have is when you put another cell type in, say, the second channel, and say we have an endothelial monolayer in one channel, a gel region, and then a different cell type over here, these cells always want to tend to migrate into the gel, and they tend to intermingle with the vascular network. And that may be what we want ultimately in terms of if these are pericytes, and they might need to stabilize the vasculature. But in other cases, we'd like to look at just the chemical signaling and not have just have juxtacrine or paracrine and not juxtacrine signaling. So in this case, the cells are actually put in these beads. These are alginate beads, so it takes a little while for the cells to get out of the beads. And during that time, they're signaling with the endothelial cells that would be plated in the region here. And kind of results, so this is the system in a little bit larger view. Here are the beads that, actually on the right-hand side here, we have alginate beads that don't contain cells. On the right-hand side, we have alginate beads that contain the fibroblast cells. And by watching this then over time, these obviously are secreting factors that are influencing the endothelial cells that are plated on the center. And what you see is, again, you can see a gross asymmetry between the vessels that sprout into the gel on the left-hand side where the VEGF plus other growth factors are being secreted, as opposed to the right-hand side where the beads are empty and you're not getting any secretion. So it's an experiment that allows you to have kind of an internal control. Okay, on one side you have the cell secreting factors, and the other side you have sort of a control case. What we're interested ultimately in being able to do is develop these systems that can actually be perfusable. Some work from Joe Tien's lab over at Boston University, they've actually engineered these vessels. And what they do is they take a block of gel and they cast in that gel channels, which they can then seed with endothelial cells. And those endothelial cells then form a confluent monolayer around the channel. They tend to be a little bit larger than the vessels that grow naturally. So here's 100 microns. So this vessel probably has a diameter of about 70 microns or so. And you can see it's lined with endothelial cells. This is actually a network that's been formed. I think this was from Strook's lab at Illinois. But in either case, you can actually form these vessels. You can check their permeability. In this case, a fluorescent dextran has been introduced into the channel, and it's diffusing outward. If you add thrombin, what you see is that the vessels become much leakier, so that much of the dye leaks out into the surrounding tissue. And you can also do studies of looking at adhesion of lymphocytes, for example, or leukocytes to the vascular wall. So here the leukocytes are fluorescent. They've introduced TNF-alpha, and the TNF-alpha tends to promote adhesion of the leukocytes to the vessel wall and also transmigration. Whoops. Here's a nice paper recently from Nuli Jung's group in Korea. And what they've done is they've produced a microfluidic system that has these regions of gel that span between two channels. Similar to what we do, they can introduce endothelial cells. In this case, they introduce them to, they have the endothelial cells on one side, typically either fibroblasts or endothelial cells on the other side. And then they can actually watch these structures grow through the gel. This is a fibrin gel. And eventually anastomose at the center in order to form a lumen that goes all the way from one side to the other. Okay, so it's a little bit of an engineered system, but at the same time the vessels are growing in a more natural way than you'd have otherwise. In our system, we've done something similar, but this is in an earlier version of ours where we had a number of PDMS posts, sort of rows of posts, that would help to stabilize the gel. This was before the students in the lab developed the technique so that they could introduce the media and control the pressures much better so that the gel didn't get pushed out, ruptured and pushed out of the region. So these were put in order to help stabilize the gel. But it turned out that the presence of these seemed to actually help promote the formation of vascular structures that went all the way across. So this is one channel. The other channel is just up here. You can just about see it. This is a confocal image of this region. And what you can see here is that you're getting nice, well-developed luminal structures. They extend along the channel like this. In fact, we could perfuse microspheres from one side to the other. And notice the dimensions here. Here's 100 microns. So some of these channels are getting down to capillary size, probably on the order of about 10 microns. So we could send microspheres of a few microns through the system. They'd flow all the way through. And we didn't see how long we could actually maintain these, but they would develop over the course of about a week. In this case, 6 or 7 days. We could also simply seed the cells in the gel, and over a period of time, these vessels would grow. This is in 5 to 10 days, and here you can see the well-developed structures that go all the way from one channel to the other channel. And again, these could be perfused, and we didn't seed other cells in the matrix, although in principle we could have. One of the other things that we've been interested in, see on our list here, we've got under gradients, we're interested in being able to produce gradients of hypoxia or maybe just a hypoxic environment. Most cell culture is done sort of under normoxic conditions. And if you want to do experiments in hypoxia, which is probably the situation that you have in tumors and in a lot of organs, you have to use a hypoxia incubator, which is, it's a massive thing, okay, which costs $15,000. And really, I think, limits your capabilities of being able to do experiments on hypoxia. Well, you can do the same sort of thing on a micro device. And the only difference here between the devices that you guys are using and the device that I'm showing here is the fact that we flow gas through channels that run parallel to the media channels. So you can imagine, for example, we might flow pure nitrogen through both channels to produce hypoxia. Or we might flow oxygen-containing gas on one side, oxygen-free gas on the other side, and the cells that would reside in the middle would see actually a gradient in hypoxia. And that's something that you couldn't produce in a hypoxic incubator. And in fact, this would be a much more stable situation because we could keep the flow going as we're imaging the cells. So this is the system in schematics. This is the computational model of what the concentrations, the oxygen tensions would be as a function of location in the device. Keep in mind that this is a device in PDMS, which is permeable to gas. Okay, we couldn't do this in hard plastics. And by virtue of the fact that it is PDMS, the gas is diffused through and you can actually then produce a hypoxic condition. This is just a mapping of, you can think of this as the flow rate of gas through those channels, and this is the flow rate of medium, and this is the partial pressure that exists in the center of the gel region. And you can see that we can get down to about 3%, maybe 2 or 3%, over quite a wide range of conditions, both corresponding to relatively high gas flow rates. Okay, but these are high flow rates in terms of like milliliters per hour. So it's, a tank would last you, one of these large tanks would probably last you about a year at these flow rates. The media flow rates, again, these are microns per minute or hour. I think these are microns per minute, okay, in order to get you up in this range. Peckley number, remember, is the ratio of convective transport to diffusive transport. So basically, the Peckley number gives you a measure of what the flow rate is. And then we've checked this out. We've done experiments to validate it. So the simulation corresponds to these solid lines and the experiment corresponds to the boxes. And depending on the diffusion, we actually introduced a film just above the region because there's too much gas diffusing out through the top of the device. When we put that film in, we get good agreement between the measurements and the numerical simulation. So we're now in the process of starting to test this. And here we're looking at migration of tumor cells. The light line corresponds to normoxic conditions. The dark line corresponds to hypoxic conditions. And here again, we essentially reproduce what people have observed in hypoxia incubators in terms of the tendency for tumor cells to migrate more actively under hypoxic conditions, under normoxic conditions. So you can imagine, again, we can start to introduce drugs into the system to see whether we can control or inhibit the migration of these tumor cells. I'm doing just like everybody else. I'm running into the question period, so let me quickly finish up. Here, this is work, you recognize this guy. Bill looked at these MDA MB231 breast cancer cells that were seeded in the gel region. The device has a slightly different geometry, but it's the same concept, introducing a higher pressure on one side than on the other side in order to introduce a flow through the matrix. So here's a pattern of the flow velocities that were computed by a finite element model. These are the cells that are seeded in the collagen gel. And here are the results that Bill found, which were kind of interesting. We thought the first thing we would do is to reproduce some results that were published on the effects of interstitial flow on tumor cell migration. Results that were published by Melody Swartz, where she found that as the flow passed a cell, that due to autocrine signaling, in other words, signals that were emitted from the same tumor cell and then sensed by that cell, that gave rise to polarization of the cell and caused the cell to move in the downstream direction. And she called this autologous chemotaxis. One more term to remember. So we set things up, and Bill did the experiments, and what he found was exactly the opposite. That instead of migrating downstream, he found that the cells were migrating upstream against the flow. So it took us a while to sort things out, but Bill discovered that, through various means, that this is a good way of illustrating what's happening. This is actually the GFP vinculin distribution. It's an antibody stain for vinculin. This is the flow direction. Here's one tumor cell, and you can notice that there's more intense staining on the upstream side, and this is just the quantitation of that. And what's happening is that as the flow goes by the cell, there's a force being exerted on the cell. And you can imagine there's both a drag force and there's also a pressure force due to the pressure gradient in the gel region. So these two combine to essentially push the cell in the downstream direction. So the cell has to interact with the surrounding matrix to keep it in place, so it's sort of holding on to the matrix, and the wind's coming at it this way. So those integrin attachments, again, are experiencing force, and they're causing activation of signaling pathways inside the cell. Okay, one of the things that signals the fact that you have forces in focal adhesions is recruitment of vinculin, which you saw from Mike's talk. And these are some of the quantitative results that Bill found. If in the case when he has flow going from left to right, this bar indicates the case where you have flow and you've blocked this other mechanism that Melody Swartz found with the CCR7 receptor. So if you block this autologous chemotaxis, you get very strong migration. That is a measure of directional migration. Very strong migration in the upstream direction. The control case, and this is without the CCR7 blocking agent. So in that case, the autologous chemotaxis in this particular instance tends to win out a little bit, and the cells tend to migrate in the downstream direction. And this is just the control. I got that right, Bill? Okay. So that's what Bill's been up to, and that led to a paper in PNAS in 2011. Finally, just something very quick on stem cell differentiation. So remember, getting back to the first part of my talk, what we're interested in doing is taking these embryonic bodies or collections of stem cells, differentiating them into different types of cells, using these cells for, say, sensing, myocytes for actuation, endothelial cells for providing nutrients to the biological machines, And then ultimately being able to use 3D printing or some way of organizing these cells in such a way to produce some sort of a three-dimensional structure. And it turns out that mechanics plays a role in virtually every step here. In terms of the effects of substrate stiffness, you saw some of that from Adam Engler's work primarily, but also other papers have come out since looking at the effect of differentiating stem cells into different types of cells by just growing these cells on different stiffness substrates. The effects of cellular tension, for example, on myocytes, some work that was done in our lab, by stretching the stem cells or getting them to partially differentiate and then put them onto a flexible substrate and stretch them, you can help to promote differentiation. And then finally, the effect, some people are looking at the effect of shear force as well, particularly in the case of endothelial cells, because the thought is that endothelial cells normally exist in an environment where there's shear stress exerted on them, and that probably would be a factor that would be important in differentiating these cells into functional endothelial cells. And then finally, Sebastian, this guy should look familiar, has come up with a really clever idea using the microfluidics to start to control many of these different factors that we identified here. So here's the gradient that you guys established. So this is the device, FITC dextran gradient, and here's the concentration measure. Think about combining this with a situation where you have an embroid body that sits inside a gel, and then you've got another channel above that is that you can control the pressure in, and it's separated from the gel region through a thin membrane, a thin PDMS membrane. So you could apply then, once you increase the pressure inside this chamber on the top, you can apply compressive stress, which would apply forces to the embroid body, not in a very detailed controlled way, but in sort of a globally controlled way. So we can do that. You could combine this with hypoxia. You can set up gradients. So if you go down the list here, we can do chemical gradients. I haven't told you about it, but we've done some experiments where we generate temperature gradients. Pressure gradients, oxygen. We haven't done pH, but that's similar to how you would do the chemical gradients. Forces, either mechanical compression or interstitial flow. Separation or organization of the cells. We can plate them into these different regions so they can actually communicate in a more natural way. We can use different matrices. We can orient the cells in different ways depending on polarization of the cells, and we can do co-culture. So I think with microfluidics, I think there's a real opportunity now to do a lot of experiments that just were not possible, even 5 years ago. So I'm going to stop there. Went a little bit over, but I think there's still time for some questions. Just wanted to acknowledge the people in my group, both here at MIT, but also in Singapore. Some of the collaborators and the funding, especially the funding from EBICS, from the NSF. So I'll stop there. I'm happy to answer any questions you have. Yeah.
What's the size and time scale of the cell-cell interaction that you see? Like, in other words, is there like a minimum distance between the cells that they feel each other, or like how long?
Yeah, it's basically diffusion times. And remember that diffusion time goes like the square of the distance between cells. So anything above about a few hundred microns, the diffusion time would be days. So you probably want to have cells within about no more than about maybe a millimeter separation distance. So in the systems that we have here, millimeter is sort of the maximum dimension that we try to aim for. And then the time scales are really determined by the diffusion distances and the time scales associated with that. Yes?
Do you think that the results which we get from the microenvironment we can interpret in the microenvironment, because normally the cells are in macro environment, not in the micro?
Well, I think because of the fact that, a cell here isn't really going to sense what a cell is doing even 2 millimeters away, because the fact that whatever this cell secretes, it's going to be so diluted by the time it gets here. that the cell is really probably interacting primarily with its near neighbors. And certainly in terms of these like pattern development experiments that people have done, the cell is generally acting with its right adjacent neighbors. So although, yeah, we're only reproducing really sort of the local microenvironment, I think it's probably not too different from sort of the more macroscopic situation that you have in vivo. One exception to that is the case where, factors obviously are circulated throughout the body by the blood supply. And so, what happens in the liver can affect what happens in the brain or a bone or other tissue through the circulation. And none of that is being reproduced here. So that, in that sense, I guess, that macroscale type of interaction, you can't easily control, but you can a little bit because, you can control what comes in through the channels. So if you want to say, well, I want to mimic interaction between, say, a liver and a heart. Well, on a chip, you could have sort of hepatocytes and sort of a liver-like culture over here. You could have cardiomyocytes and a heart-like culture over here, and you could have a channel that connects the two. And you can do onboard pumping, so you can actually circulate media from one device, from one region of the device to the other. So what people are doing now is they're developing, you've heard about organ on a chip. Now people are developing sort of human on a chip models that are being supported by like the FDA and NIH, the development of these things for drug screening. So you'd have heart, you'd have muscle, you'd have liver, you'd have lung, and all of these would be interacting and communicating with each other. So then you'd really start to include the effects of the circulation as well. Yeah, maybe. Yeah.
I was wondering if, and you might have mentioned this, if you could use something besides PDMS, especially if you could create it on a medium where you could create your angiogenesis pattern or whatever and then dissolve that out and sort of have like a freestanding structure.
Yeah, in fact, Sebastian's been playing with one way of doing that. I mean, one way to do it is to use different types of gels. And some gels, for example, like the alginate, you can dissolve with citrate. So you can form sort of a two-gel system and then dissolve away the agarose with the citrate. But the way that Sebastian's been doing it is taking the systems like what you see here, and rather than coating the surfaces with PDL so that they're sticky to the collagen, leave them uncoated. And he's found that he can actually peel off the PDMS and leave behind the gel regions, with all the structures and everything intact. So you can imagine, let's say we want to have multiple layers of these things. You could do this in multiple systems, peel them off, and then pile them on top of each other. So I think there are ways of doing it, and then also, the inkjet printing ideas that people are using. They printed everything from single cells to embroid bodies. So I think those technologies are really just emerging, but I think it's something that we'll see probably more commonly in the next couple of years. Yeah.
In the system, I think you have considered the effect of shear stress due to flow on the endothelial tissue, but I would imagine when blood is flowing through the blood vessel, the endothelial tissue is also being stretched out. So how would cyclic stretching actually affect some of the processes? For example, cancer cells, say, invasion into the tissue?
It's a good question, and some of the devices that people are making, we're generating one. We've used one actually in our system too. You can also grow the cells on a substrate that stretches. So, for example, there was a nice paper in Science about a year, maybe 2 years ago now, from Don Ingber's group across the river, where it was a lung on a chip model. And in their case, what they did is they set it up so that they had two air channels, one on either side of the sort of like this. Okay, so this is the device. Okay, this is air. This is air. This is the thin membrane. And they could plate cells on the top here. So for example, airway epithelial cells or alveolar epithelial cells, they could plate a different type of cell down here. This membrane was porous. And by changing the air pressure here, you could periodically stretch the membrane. Okay? And this was, on the order of about millimeter scale. And they could have, they would have fluid down here then, and air up here. So this would represent the gas portion of the lung. This would represent like the blood supply, the capillaries in the lung. So there are ways of putting that in. And it's recognized that the stretch is very important, but it's a little bit more complicated. But certainly, within the capabilities of things you can do. Yeah.
Yeah, I have a question about the potential differentiation on substrates. So when the substrates have different stiffness, like what are the cells sensing?
There are different theories about that. How are they sensing the stiffness? We think that what's happening is that there's the actomyosin machinery inside the cell, and that it's always trying to contract the cell. If the cell is on a very stiff substrate, you can't get much strain, you can't get much contraction. And what happens is you have the myosin motors trying to walk along the actin filaments. They don't walk very far before they reach the limit of how much force they can generate. Okay? So they tend to generate sort of the maximum amount of force that the motors can generate in a stress fiber on a stiff substrate. Okay? And no matter how stiff you make that substrate, then it's not going to make any difference. So the forces internally internal stresses tend to plateau. If you're on a soft substrate, these motors can actually go quite a ways because there's a lot of compliance, so the stress fibers have to contract quite a bit in order to, before they reach the limit of what the motors can generate in terms of force. Well, what happens is as the motors walk along, they become sterically impeded. So in other words, they're walking along an actin filament, and then they'll reach a site that's already occupied. So the motor gets stopped, it gets stalled, it's jammed. And as a result of that, and when we've done numerical simulations of this, as a result of that, the forces that can be generated, the stresses internally that can be generated, start to fall down. So in fact, there's almost a linear drop in force from this plateau region down to zero as the substrate stiffness gets lower and lower. So the way then that we think that a cell would sense force is that, that's how the different stresses are generated. So you can imagine that if you're only halfway up this curve in terms of the stresses that are generated internally, well, that means that the proteins which are experiencing the force or transmitting the force are sort of at a midpoint in terms of the amount of force that they're actually having to support. Therefore, that's going to limit the amount of conformational change that they would experience. Okay, so that's going to determine then the level of signaling that they can actually either activate or the conformational change that leading to recruitment of proteins or things like that. So that would be one mechanism, sort of a graded sensing by these force transmitting proteins, a mechanism by which you could control the different signaling pathways. Yeah.
Can one measure the force of lifting by that microfluidic experiment?
A force of what?
You showed the thing where you can apply the force from the bottom and lift the cell.
Oh, yeah.
So is it measured by the how addition force of the say to the substrate and all these things by?
So the way that those forces is due to the flow from the bottom coming up.
Yeah.
And I mean, we can certainly control it, but what was your question then?
What's the lifting force? Basically the
Oh, what is the force? Okay.
addition force.
It's on the order of about 50 to 100 Pascal. And if you integrate that over the total surface of the cell, you're talking about a few nanometers per cell. I'm sorry, a few nanonewtons per cell. So it's certainly within the range of what, it's comparable to the stresses that we know elicit a biological response from the cells due to shear stress.
I just want to add another experiment talking about microfluidics. In 2005 in Nature there was an experiment on a microfluidics. One embryo is kept, and from two sides two fluid was flown at different temperature. And embryo development processes happen very much sensitive to temperature, how slow and how fast it happens. And it is observed that part which is warmer, the embryo development happens much faster than the other part. But after some time, that part gets stalled, so that this part also balances that stage, and then embryo develop happens normally. So,
well, that's interesting. I didn't see that.
Yeah, it is a 2005 Nature one paper on microfluidics.
Do you remember how large a temperature difference they produced?
12 degree and 35 degree, around that. In that whole range they did. So they have seen even the gene expression and all the pattern. After some time it is constant.
Yeah.
So they control between anterior and posterior side in Drosophila embryo.
Mm-hmm. I should take a look at that. Anything else? I think it's lunchtime.